So for the next presenter, it's going to be Nurudeen Rahman, who is currently in Switzerland at the Swiss Tropical and Public Health Institute, and he's going to take us through nanopore-based analysis, unravels the phylogenetic placement of human-infecting Trichuris species in Cote d'Ivoire, as well as Laos, Tanzania, and Uganda. Welcome, welcome. You can take over. I think your audio is off. You can put it on and then continue.
Okay, can you hear me now? Perfect.
Yes, yes, we can hear you now.
Thank you very much, Emmanuel. Hello, my name is Nurudeen, and I'm a PhD student at the Swiss Tropical and Public Health Institute, an affiliate institute of the University of Basel. And I'll be happy to give a talk today on nanopore-based analysis unravels the genetic differences of human-infecting Trichuris species in Cote d'Ivoire, Laos, Tanzania, and Uganda. So, firstly, I would like to this move. Yeah. So firstly, I would like to tell you a bit about trichuriasis, an infection caused by Trichuris trichuria. It's one of the major soil transmitted helminth infection, infecting over 500 million people worldwide, with heavy infection leading to severe anemia, growth retardation, and cognitive impairment in children, which leads to huge socio-economic development burden on people living in affected areas. Control of these infections has involved the use of drugs in mass drug administration ways, like benzodazoles, which includes albendazole and mebendazole alone or in combination with ivermectin in endemic areas. However, in context, these drugs alone, which is, benzodazoles alone, actually very good and very efficacious when it's used against other soil transmitted helminths like Ascaris lumbicoides and Ucum. However, in the context of trichuriasis, these drugs are not as efficacious as they should be, which brought about the use of a combination therapy or a combination of drugs with ivermectin, which now increases the efficacy of those drugs. So this genus Trichuris, they are very elusive species, and quite a few studies have suggested that these species are kind of excellent candidates for hiding cryptic species, which means that this genus have species that are kind of indistinguishable using the morphological analysis, which means you can actually see them on the microscope, but then they look very much alike, and you can't decipher the differences between these species. So most times, what we actually do most times is actually to look at doing more of ribosomal or mitochondrial markers to actually differentiate the differences between these two species. And these papers have suggested that majorly there are two clades of trichuriasis infecting humans. So for our study, we focused on the randomized control trial, which is conducted in 4 regions, comparing the efficacy of albendazole monotherapy and combination treatment with ivermectin. So this study was carried out in Pemba Island, Laos, Cote d'Ivoire, and Uganda in 2023. So like I said earlier, in context, everyone knows that this drug, ivermectin and albendazole, works quite well for trichuriasis infection, but we noticed the kind of reduced efficacy for this drug in Cote d'Ivoire, which actually just brought out a kind of light, like what's actually happening. So what we saw then was that there was a reduced efficacy in Cote d'Ivoire with a cure rate of 14% when compared to other countries that we did our trial, Cote d'Ivoire, Laos, and Pemba, which had a higher efficacy for these drugs. So what we tried to do after our trial was to send a subset of this sample to a collaborator we have in Calgary, and they did an amplicon sequencing using the ribosomal and mitochondrial markers for a set of samples we have over there. So then we got to know that this trichuriasis infection actually have low sensitivity to these drugs, because we have a novel Trichuris species actually kind of found now in this country called Trichuris incognita. So what we tried to do also again was to now send carry out another study in Cote d'Ivoire using a whole genome sequence. and we did a mitogenome analysis and created a tree. And we also found out that yes, these new species are actually really in Cote d'Ivoire. Then we did from the worms expulsion study we did, we found out that all the worms we exposed there were actually new species called the Trichuris incognita. So that brought me back to my study, and like, well, we need to just see how we could actually work on this samples we got from Cote d'Ivoire, Laos, Member, and Uganda. Then we tried to use a nanopore-based amplicon sequencing of the ribosomal ITS2 DNA marker to explore the genetic diversity, basically, of those drugs. In context, there has not been a reason for reduced efficacy of those drugs basically anywhere across the globe. But 4 reasons could amount to us having a reduced efficacy for drugs. And one of them is parasite resistance. But then we've never found a reason for parasite resistance in Trichuris infection, albeit we've found several infection or resistance in veterinary important parasites. And also another reason for parasites failing for treatment failure could be a result of genotypes of worms being different in the regions that the sample was carried out. So that's why we kind of talked about, okay, yeah, let's see how we could use Nanopore-based analysis of the ribosomal ITS2 to explore the genetic distribution of this Trichuris population in the 4 regions. So for these, we used our starting materials, which is stool sample to extracting stool samples, which were in ethanol for the samples gotten from Uganda, and frozen stool samples from Laos, Member, and Cote d'Ivoire. We also included an additional optimization step, which includes freezing, beating, and eating to lyse the eggs of the Trichuris eggs properly, following which we now extracted the DNA using the PowerSoy DNA kit. So once this DNA has been extracted now, then we use the barcoded ITS2 primer pairs to produce barcoded amplicons, which were pooled, quantified, and sequenced at depth using the Nanopore MK1C platform. Following these, the reads were kind of base called and demultiplex using ONT barcodes. And also, if you remember, I told you we use we used primer barcodes, which means we also have to demultiplex this reads using the PCR primer barcodes. Following that, we now did a sequence analysis. So for our sequence analysis, we used Data 2 pipeline for amplicon sequencing analysis to sequence this demultiplex raw reads based on quality filtering, length filtering, and also denoise the reads to remove chimeras. Then the final output generated was a table containing amplicon sequencing variants for each of the individual samples we have. We also included an additional filtering steps to remove chunks or off targets, because we know we worked with stool samples, so we expect to have bacterial or fungal contamination in there. So following that, the filtered reads were aligned using MAF2 for multiple sequence alignment, following which these aligned sequences were visually represented using clustered ASVs at 97% identity. After which haplotype networks were generated using the pop hat, and we kind of produce maximum likelihood trees using RAXML. So moving on to our results, here we can see that we successfully generated about 93% of amplicons from our samples. From 670 samples we worked on, we generated from 626 samples, and the table below shows the results from our capillary example of results generated from our capillary gel electrophoresis with the bands at 592 base pair, which was our where we are expecting the bands for our ITS2 basically. So following that, we had to do analysis, the analysis of the ITS2 marker, which involved 3 different analyses. First, we did an haplotype network, which shows that 2 different haplotype groups are actually present in these 4 regions, which one of them is actually dominated by those from Laos, Pemba, and Uganda, and the other one is being dominated very well by the Cote d'Ivoire population, showing a clear genetic divergence of 2 Trichuris haplotypes in these 2 regions. However, it's what's noteworthy that this different genetically divergent Trichuris species are also present in the other countries, albeit in less amount. So Following that, we did produce the maximum likelihood tree to include to also include curated ITS2 full-length sequences from publicly available ones in GenBank, which includes ITS2 sequences from rodents, from livestock, from non-human primates, from suis, which from pigs or porcines, and also from humans. And also we actually had to extract from the whole genome sequence, we had then extract the ITS2 from our group. We had the whole genome sequence. We had to extract ITS2 from the whole genome sequence we have in our group and also added it to the tree, so it makes more sense that way. So what we found out was that majority of these haplotypes from Cote d'Ivoire clustered in a clade separate to most of those found in Pemba, Laos and Uganda population, with the clade also containing some non-human primates species, which are also very close to Trichuris suis, which is the Trichuris of pigs. This also agrees to the preliminary reports I talked about earlier, which said that we had a new species in Cote d'Ivoire. However, we also observed that these new species are actually present also also present in also the other countries, which is Pemba, Laos, and Uganda, but in minute quantities or with low prevalence, I would say. So then we also now performed a pairwise percentage identity calculation, which also alluded to the fact that majority of the samples from Cote d'Ivoire are also found in the clade D, which is the clade containing the Trichuris incognita, which is the newly found Trichuris species. So then now we could actually say that yes, this Trichuris incognita is present in the 4 countries which we sampled from, and not just only in Cote d'Ivoire. So in the course of our study also, we noticed that fragment length is actually a kind of important part of this process, because we noticed that the fragment length of this known Trichuris species of human, the Trichuris trichuria, had a 520 to 540 base pair length, and the one from the newly identified Trichuris species is having base pair length of 580 and 592 length. So what we tried to do was to look at the fragment lengths of all species of Trichuris that we know of, and we observed a signal difference in fragment length between all clades, except between Trichuris muris, which is Trichuris of rodents, and the one of Trichuris of livestock. So then we evaluated the predictive power of ITS2 region fragment length to discriminate the differences between the 2 human-infecting Trichuris species, and our result actually showed that the fragment length is an accurate diagnostic marker to differentiate between the 2 human-infecting Trichuris species, which achieved a classification accuracy of 95.4%. So in conclusion, we actually found that haplotype network and phylogenetic analysis revealed that morphological traits alone are insufficient to distinguish the closely related species of Trichuris trichuria and Trichuris incognita. And also we found out that Trichuris incognita is actually present in also other countries apart from Cote d'Ivoire, which is Laos, Uganda, and Tanzania, albeit in lower quantity. Also, we found out that this Trichuris incognita is closely related to Trichuris suis, but it's not a zoonotic infection as found out by a work done in our group, which puts in the context of whole genome sequence samples exposed from pigs and humans in Cote d'Ivoire. Then this newly formed Trichuris species called Trichuris incognita is entirely related to Trichuris trichuria. Finally, we used fragment length to decipher that yes, it's a robust diagnostic marker. We identified it as a potential PCR-based diagnostic marker, which could accurately differentiate between these 2 human-infecting Trichuris species. And this could, in a way, eliminate the need for expensive sequencing platforms and establish a cost-effective and robust method to delineate the differences between these 2 Trichuris species. So I would like to acknowledge the team from Calgary, which helped us with the subset samples at first, then the team of researchers that we worked with in Laos, Pemba Highland, Uganda, and in Cote d'Ivoire. And also my team, the HTDU team in Swiss TPH for the assistance. Thank you.